Good afternoon and welcome to ABG Sundal Collier's pre-IPO seminar. My name is Adam Carlson. I'm an analyst in the healthcare equity research team. Today we are hosting 7 companies who will be presenting in half-an-hour slots up until 4 p.m. 20 minutes worth of presentation and about 5 minutes worth of Q&A at the end. It's my pleasure to welcome the first company, XNK Therapeutics, and I'll hand over the word to CEO Johan Living at XNK.
Thank you very much, Adam. I'm very pleased today to present XNK Therapeutics to you. XNK Therapeutics is an immunotherapy company focusing its efforts on developing novel NK cell-based therapies. And NK cells are natural killer cells which are part of the innate immune system. We are today a world leading in the field of autologous NK cells, and autologous meaning using the body's own cells. And we, of course, are working very hard to keep and preserve that global leadership that we have today. So next slide, please. This is our disclaimer, and you are, of course, feel free to read that whenever you have the time. My name is Johan Living, and I'm the CEO of XNK Therapeutics. I joined about a year ago. At the core, I'm an engineer and also an economist, and I've been working the last 13 years at Janssen. The last 5 years, I worked globally, and Janssen is the pharmaceutical company of Johnson & Johnson, the world's leading pharmaceutical company. The last 5 years, I worked globally in a team that was trying to look at the R&D portfolio and saying, how can we commercialize these assets? And at the same time, looked at the region and saying, what is it that you would like to sell? And basically trying to make a fit in between those two things. And I learned a lot, and hopefully I can bring a lot of that experience into this company and what we are currently doing. I have a personal interest in the research of multiple myeloma, a rare blood cancer, and was basically handpicked by the team behind this company to take this to its next step based on my global pharma experience and research in multiple myeloma. Next slide. If we take a look at XNK Therapeutics and why we believe this is a very good investment case. First of all, is built on the world's leading research on NK cells that is going on here at the Karolinska Institute. It was here in the mid-70s where the function of the NK cells were discovered, and today this is one of the world's leading research center when it comes to the research around how those cells work. And at the core, at our company, we have a platform, a technology that would allow us to expand and activate these NK cells. And by expanding, we mean multiplying them, making them many, many more, and activate, actually making them cytotoxic again, we have found a way how to make them want to kill their target cells, their cancer cells again. This platform is both stable and versatile and will allow us to do many, many things. We come quite far in the development and the clinical development of the platform and its technology, and we have passed phase 1/2 study that we presented a year ago, showing good safety profile and a promising efficacy. We are now ready to take the next step, and before the summer, we will start a phase 2 trial in the same indication, multiple myeloma, in combination with an anti-CD38 antibody, called Sarclisa from Sanofi. So we're going into collaboration study together with Sanofi and the Karolinska Institute. If you look at the potential of this company, the price points that you can take are very high. And we have an effective production, and we can get a very, very good margin. So the price points are high, and the patient populations are quite large. So for these types of products, of course, the potential will be quite big. Next slide. We are not a new company. The company was founded back in 2011, and it was founded as a spin-off of the research that's been going on here at the Karolinska Institute. And the foundation was a research grant from Vinnova, which is a Swedish research foundation. And the whole concept was to trying to make a product, trying to commercialize the research that has been going on NK cells. Back in 2011, the interest of NK cell research was pretty low. The focus were on T cells, another part of the immune system. So the company could work in a relatively comfort zone and could develop the technology without having too much interest in the company. However, that has changed dramatically over the last 2-3 years. So we have developed the platform in an academic industry collaboration fashion. We have doing that through grants, and we are currently developing our current platform in a collaboration called AdBioPro, and we're a proud member of the next big consortium here in Sweden called NextGen NK, who is trying to find the next generation of these therapies. That development has given us a patent portfolio. Today we have 3 patent families, and hopefully we'll soon file for a 4th patent family. The regulatory pathway also will come a bit further. We have an orphan drug designation that would allow us market exclusivity in both the US that we got back in the EU that we got back in 2018, and in the US that we got in the fall 2020. And as I said, from the clinical trial perspective, we started our first clinical trial back in 2015, a phase 1/2 clinical trial, and we presented that data last year at EHA. And we are now ready to start our next big clinical trial, which is a phase 2 trial combination with anti-CD38 antibody. When I joined the company a year ago, it was a clear mission. It was a rapid development in the NK cell field, and this company wanted to maintain its leadership position in the autologous NK cells. So clear mission, and we just get going. We brought in money to be able to do that in April, 64 million Swedish krona in a private placement, and we just built up and announced our management. I'm extremely proud to have Johan Erskan joining us as a Chief Medical Officer. Johan has a background in hematology here at Huddinge. He's been treating quite many patients. He has been leading the clinical development today from melphalan for oncopeptides. And previously to that, he was working at Janssen when I was dealing with Janssen's anti-CD38 antibody, daratumumab. So he has an ideal background for joining us. At the same time, Professor Mikael Uhlin will also join us in the summer. Mikael is today a professor at KI in gamma delta T cells, but he also head of the clinical immunology here at Karolinska University Hospital. So with those 2 people coming on board, I believe we will have a dream team. Next slide. The current management that we have, and which we will then, of course, will expand with these people, is Evren Alishi, who is the founder of this company. He is today one of the, I would say, top 10 NK cell researcher in the world. Vera Franzén, who is helping us from the beginning with the regulatory pathways. She has a background from the Swedish MPA and has also worked within this field of cell and gene therapy for the last 10 years. Harriet Nahi, our clinical advisor, who was the PI for our first clinical trial, is today one of the leading hematologists in the Nordics. He is a former president of the Nordic Myeloma Study Group and probably the one that treats the most myeloma patients in Northern Europe. We also have a very seasoned CFO, Hans Eriksson, with a background at Ericsson and Elekta. And our current head of R&D, Stefan Mjenke, is also a very good NK cell researcher. And Marcus Lagerlöf, who many of you might know, has been working with the company for the last 5 years, working out our business development function and trying to position this company in a strategic way. Next slide. So we are working on natural killer cells, which are part of the body's innate immune system. And they were discovered here at the Karolinska Institute in the mid-1970s. And how these cells work naturally is your first line of defense to virus infections, but also to cancer development that naturally occurs in the body. And they actually work by recognizing cells that are coming from your own body. And they do that by every single cell that you have in your body express a MHC complex or MHC receptor. And the NK cells, if they see these receptors on the cells, they say, this is a cell that belongs to my body, and I will not kill it. At the same time, you have ligands on the cell surface that's trying to activate these NK cells, and as long as they are in balance, nothing will happen. If you have a virus infection, they don't have these MHC receptors, so the NK cell will say, okay, this is a cell that doesn't belong to the body, and I will kill it. The same thing goes for cancer cells. When they start to grow quite rapidly, they lose these self receptors, but they also will start to overexpress these ligands that activate the NK cells. And so the NK cells will recognize that this is a cell that doesn't belong to the body and will kill it. And this is a process that is going on naturally in your own body. Of course, we started with multiple myeloma, but there is nothing that would say that this wouldn't work for other cancer types. So our platform technology is basically we can scale it to multiple cancer indications, but we started with multiple myeloma. What really got me excited about this company is the development that has been going on in the NK cell field. So next slide, please. 2 years ago, when I was sitting at Janssen, before that, everybody was talking about T cells and the CAR T cells and the development of the T cells and how that would work. And then it was starting to bubble. People were starting to talk about NK cells, the part of the innate immune system that we are in. And there were some really cornerstone or major clinical trials that was coming out showing that we can actually get this to work. The NK cells will work, and the side effects will be quite different. Basically, the side effect profile that you will see on NK cells they're very few, and they seem to be very good to the body. They're much more targeted killer. So there was a lot of interest going on, and since about 2 years ago, the whole field has literally, exploded. There's been deals after deals after deals, and the development is very, very rapid. The good about this company is that it was founded 10 years ago. So that, due to its history and its pace and, the collaboration with the Karolinska Institute, it is in a very good position to participate in this race going forward. And today we are the leading company in the field of autologous NK cells. Next slide, please. The business model is pretty straightforward for a company like this. At our core, we have our platform technology. That platform technology allows us to expand and activate, multiply and make these cells cytotoxic towards different types of indications. That platform can be reiterated, so we can make it better and better and better. And every time we will make it better, we will have a new product. We have taken that platform technology further in its clinical development in multiple myeloma, and we passed phase 1/2 and are now ready to go into phase 2. That doesn't say that this platform wouldn't work for other cancer indications. We've currently screened around 70 other cancer indications and looked for the scientific rationale. If we can combine our product with an antibody, so we can work through antibody-dependent cytotoxicity to get synergistic effects, and also look for strong business cases. And hopefully in the near future, we'll come into a position where we could announce the development of further indications. We are today the leading company in autologous NK cells. There are a lot of buzz going around allogeneic NK cells. And of course, allogeneic cells has an advantage of being off the shelf. So you can basically produce them in advance. And then, of course, when the individual gets sick, you have the drug ready to go. That's not possible with autologous because then you need to produce it when you actually have the patient in front of you. So you will have a different problem with logistics. However, autologous cells has some quite advantages to allogeneic one. First of all, the route to market, at least for our NK cells, will be much easier because it's not a transplantation. So the safety profile will be viewed differently from the regulatory authorities. Secondly, it's not a transplantation, so we don't have to immunosuppress the patients. So we can treat them with our NK cells without a preconditioning. Well, that would allow us to go after different target groups that the allogeneic cells really can't reach. Another interesting thing is that since it's an autologous product, we can give multiple dosing because we don't have to immunosuppress the patients. That means we can treat them, we can retreat, and then we can treat them again. And that is, of course, a clear advantage for a therapy. It can also be readily combined with these antibodies, which of course is optimal if you want to have synergistic effects. And based on our portfolio, since we've been such a company but that been around for so long time, we have a very long stability in our products. We can keep it in the freeze for almost 10 years. Next slide, please. If we look at the platform that we have, it's a pretty straightforward process. It starts by the patient donating 1 unit of blood, that's 450 milliliters. That blood is then shipped to us, where we take out our starting material and put that into the freezer. And we can then freeze and store that starting material until we feel we have the production line ready. Then we thaw it and start our process, and that's the third box there. That's a 15 to 21 day process where we expand and activate these NK cells. And it has to take some time because these cells will not really become cytotoxic before the day 10. So in this process, it takes some time to restore the functioning of the NK cells. After we have completed the production, we will freeze the product again. And as said, we have around 10 years stability data on this product. After that, the frozen product is shipped to the patient side where you can give the dosing repeatedly. And all this is being taken care of is open care. So we don't have to put in the patient to intensive care unit or whatever. You can give this in the open care and the patient can literally go home the same day. The process is closed and automated, which of course is important if you want to be able to scale this. And we are proud that we think we've done this from the right way back from academic research when we were doing the first steps back in the mid 2000. The platform has given us, and the development has given us a couple of patent families. Next slide, please. Today we have 3 patent families that we filed for. It's a treatment patent that we have in the US. It's an activation and expansion patent, basically how the process works. Then we have a combination treatment patent that we combine our NK cells with a high-dose antiviral treatment. And that is an important patent because we believe that all autologous NK cells will be needed to be combined with high-dose antiviral treatment. And then we will soon file for our new and improved process. The good thing here is that we also have an orphan drug designation. We have that gives us market exclusivity both in the US and EU. But important to say when it comes to these cell and gene therapies, you cannot just take a process, even though you know how it works, without a clinical development. So you have protection by itself by being in this field because you can't transfer the clinical data. So everybody that wants to copy what we do will still have to do the clinical development. Next slide then. If we start to look at our clinical development for our process, we are working in the field of consolidation treatment. And that is the first step of frontline treatment of multiple myeloma patients. We come quite far. We passed phase 1-2 and are now ready to go into phase 2 in a combination with an anti-CD38 antibody. We have a preclinical data in smoldering, which is a pre-stage of multiple myeloma, and we know we can expand and activate these NK cells. We also know we can expand and activate these NK cells in the patient population that will not have this consolidation treatment. So basically, we can cover the entire field of multiple myeloma patients. Next slide, sir. Our first indication, what we're going for, is a consolidation treatment of patients who are undergoing a bone marrow transplant. So these are newly diagnosed multiple myeloma patients. They will come into the clinic and they will get diagnosed with multiple myeloma. They are considered young and healthy, which is basically below 70 years of age. So they will go for transplantation. And that's a package of treatments. First of all, you get an induction treatment. That's a combination of 3 different chemotherapies with the aim to reduce the cancer burden. After that, you will mobilize the stem cells and you will collect them from the peripheral blood. After that, you go in with a high-dose chemotherapy, trying to knock out everything. And by doing that, you take out all the cancer cells, but also your immune system. So you have to restart that by giving back the stem cells. At that specific point in time, all the cancer are not gone. They will still have, bits and pieces in the body that has not been taken care of. And the patient is very weak. And that is where we come in. We believe that our NK cells has ideal properties for really, destroying the remaining of the cancer cells, or as much as we can, in a very frail population where few other combinations actually can enter. And that will then go on with the whole aim of trying to really prolong the time until the patient's cancer eventually will get back. So what have we done then with the clinical trials? Next slide, please. We have done a first-in-human phase 1/2 clinical trial. We treated 6 patients here in Huddinge, and the primary endpoint was, of course, safety and tolerability, but we also looked at the efficacy. Next slide. So what is the data telling us? Well, we believe we have a very good safety profile, but also a promising efficacy. So on these patients that we treated, we didn't see any severe adverse events. The majority of the adverse events that we saw were grade 1, so they were mild, and they was being considered unrelated. It's things like upper respiratory infection, lumbago, diarrhea, these type of adverse events. There was, however, one thing that stood out, that the first 4 patients were developing shingles. And of course, we got quite worried about that. And we activated the entire network of NK cell researchers. And it actually happened that Hans Gustav Ljunggren, who also sits in our board of directors, who also a world-famous NK cell researcher, said, "Oh, I've done research in mice in the mid-1990s. And what is happening, and these highly activated NK cells will look for the reservoir of chickenpox and basically reactivate the virus, and you will get shingles. That can easily be treated by a high-dose antiviral. And so we gave this treatment to the last 2 patients, and they did not develop shingles. And we also, based on that happening, also became filing for a patent. If we look at the efficacy side, very pleased, all of our patients are still alive after more than 5 years. That's, of course, good. If you look at the historical control group, roughly 70 to 75% is still alive. So 100% is on the upper side. The progression-free survival, which is the time until the cancer comes back, are 34 months. That's on the higher side, probably 28 to 30 months in a historical control group. What I really like about the PFS is that we have still 2 patients who, after about 5 years, still hasn't progressed. Next slide. If we look at the response, the destruction of myeloma cancer cells, you have the number of myeloma cancer cells on the y-axis. And what you can see, wherever you look, we actually get down. So we will improve the response in all the patients where we can measure. And of course, that's really encouraging for us. What we also noticed that we have a biomarker, a granzyme B. When these NK cells are doing their killing, they're using perforin and granzymes, and we can measure the level of granzyme B. That's the next slide, please. And what you would see, first of all, you would like the NK cells, after they're being fused, you would like them to see them going out in the body. They should not stay in the circulation. So to the left, you see a graph showing the 3 infusions of every patient and what is happening with the granzyme B levels in the serum after 30 minutes, 60 minutes, and 240 minutes. And the dosing are escalating, which means you would like to see higher and higher peaks. And we see exactly that. Then you would like to see the levels dropping off because you would like the NK cells to go out in the tissue. And that is exactly what we're seeing. So we know the NK cells that we infuse, they're being uptaken by the body in the way we would like it to see. But what really making us think this is fantastic is what happened in the bone marrow. So we have bone marrow measurements 4 weeks after the last infusion, and we can see elevated levels of granzyme B in all the patients where we have bone marrows. To us, that tells us our NK cells are still in the bone marrow and are still active. Next slide, please. So our next step. Well, before the summer, we hopefully will start our combination trial where we are going to combine our NK cells with Sanofi's anti-CD38 antibody, Sarclisa. And that's going to give us, we think, very strong synergistic effects because these antibodies, they are using the body's own NK cells to do the killing. So if we add a lot of highly activated NK cells, we believe there will be strong synergistic effect. At the same time, these antibodies are depleting the pool of NK cells in the body, getting the patient more prone to get infected by virus. So by adding NK cells, we hopefully can take away the side effect problem that these antibodies has as well. So hopefully this trial will give us strong data on the efficacy on combination. Next slide, please. So we are also planning more trials to develop our clinical pathway for this compound, and we are actually designing a monotherapy trial right now, which we believe give us further data for our product. However, we will not stop within multiple myeloma, as I said. Next slide, please. We have several tangible opportunities to go for other cancer indications. And there we are looking for these pathways where we have an antibody that we can combine with, but also point in the treatment where we have a low cancer burden, because we believe that our NK cell therapy will have an optimal place in the treatment where you have a low cancer burden. And we are currently doing quite a lot of work to identify this. And hopefully we'll be able to out-license one or two of these indications. So to sum it up, next slide, please. XNK Therapeutics is a global leading company in the field of autologous NK cells. We are built here on the research in Karolinska Institute and taking advantages of this whole infrastructure. At our core, we have a platform technology that is both stable and versatile and would allow us to do many different things. We come quite far in the clinical development of our compound in multiple myeloma, and we're now ready to enter a phase 2 clinical trial in combination with Sanofi's anti-CD38 antibody and in collaboration with the Karolinska Institute. If you look at the financial view of these type of companies, the opportunity and the potential is very high. Thank you very much, and happy to take any questions.
Thank you, Johan, for that presentation. I think we have time for just one brief question. As you said, you've completed a pre-IPO placement. What can you say about your potential IPO plans going forward?
Well, first of all, right now we have we have a lot of money in our pockets, which is good, so we can really do the expansion as we wish, and we can really accelerate. We haven't made any final decisions on the plan for IPO yet, but we are evaluating all options.
Great. In the interest of time, I'm afraid we have to move on there, but thank you very much for an interesting presentation, Johan.
Thank you.